see there was a recent estimate of the loss due to plant viruses worldwide. That comes to some, if you work it out, like 50 rupees per dollar, you will get some 30,000 crore rupees loss per year. So that itself is one good reason why we should work on plant viruses. So to reinstate the economic importance, it's they induce disease in a variety of food crops worldwide, and the yield of the crops can reduce up to 100%. And if you take all the pathogens together, that is fungus, bacteria, virus, viruses alone cause up to 15% of crop loss. And then one major problem is viruses are very difficult to control. And what are the costs associated with plant virus infection? First of all, you have to control the vectors, like by using pesticides, etc. But you know the problems associated with pesticide use. Then the crop loss. Then you need to remove the diseased plants, otherwise the disease will spread from one plant to the other. Then quarantine and eradication programs. And finally, research and education, like paying for people like us, is also a cost. And so, what are plant viruses? These are small infectious particles that are called virions, and you need a highly powerful microscope like electron microscope to look at the viruses. And they are composed of a protein coat, sometimes there is a lipid also, and of course the genome is a nucleic acid which is inside, and typically they code for minimum of 3 proteins, and could be more also. Then the genome is the one which produces the proteins, and sometimes a nucleic acid genome can produce as much as 1 million copies of the viruses, and they are obligate intracellular parasites, that is they can replicate only inside a host cell, and they can be stable for many years outside the host. Next one. And what are the symptoms and diseases? This is considered as the earliest record of a plant virus disease, which dates back. She mentioned that even in summer, why is this plant showing symptoms? Why is this plant showing yellow colored leaf? Because usually in the autumn or the fall season only, leaves turn yellow, orange, red and things like that. But why is it turning yellow even in, why is this plant showing symptom even in summer? At the time she didn't know it was due to a virus. Next one. Okay, these are some of the common symptoms you will see, like chlorosis, that is lack of chlorophyll, and yellowing of plant parts like leaves, fruits. Next one. And mosaic symptoms, most common symptom, you can see even in tulips, this kind of symptoms, which initially people didn't know it was actually due to a virus. They thought it is a different variety. So people went in for that variety thinking that it is a novel plant, but later on it was shown that it reduces the vigor of the plant because it's actually a virus called tulip breaking virus. Next one. Then you can see the vein banding, veins become thick. Then next one. These are some of the various symptoms: necrotic spots, spread of necrosis, necrosis even in fruits, and then leaf. Next one. Then you can see the leaf and fruit distortion. See, the fruits become so distorted that we would not want to eat them. Next one. No, this is spring spots. Next one. You can see how the plant gets stunted. This is a healthy one. This is a virus infected one, and how the fruits also become deformed and stunted. Next one. And then little cherry disease, where you can see the infected cherry becomes so small. Next one. And then in plum, when there is a virus infection, fruits prematurely fall down. Next one. Okay, so these are some of the diseases I had mentioned earlier. So we can go to the next slide. How do we control plant virus diseases? plant viruses. Next one. Now coming to the genome properties. So it can be an RNA or a DNA virus. RNA can have single or double stranded, linear or circular, and DNA also single or double stranded. And then it can be either plus stranded, plus is the coding strand which is a sense, or non-coding strand or the antisense. And the genome can be split into several, one or two or three or several components. We'll see all that later. Next one please. And then this is what we'll tell you more about it. Plus sense single-stranded RNA can have 1 to 3 segments. Plus sense single-stranded RNA viruses are the most prevalent of all the plant viruses. Then other genomes also like circular double-stranded DNA, circular single-stranded DNA which can have 1 or 2 segments, and linear double-stranded RNA which can be segmented, linear negative sense, that is a complementary sense single-stranded RNA which is also segmented. So these are all various families of plant viruses which you will find. Next one. Okay, this is just to summarize whatever I showed you earlier, so we can skip this. And what are all the proteins that are commonly encoded by plant viruses? First of all, it needs a nucleic acid polymerase because as you know the genome has to replicate, whether it is RNA or DNA. With the help of the host proteins it replicates into more and more copies, and then it translates whatever code protein and various other proteins which are necessary, like helicase, movement protein which helps in the virus to move from cell to cell and also long distance along the phloem. Then capsid protein which is the one which protects the genome from getting degraded in the host cell. And protease, some of the viral genomes are translated into one large polyprotein, and this polyprotein has to be cleaved into functional proteins by viral encoded proteases. Then transmission protein, that is for insect transmission, viral proteins help so that the insects can carry this viruses from one infected plant to the other healthy plants. Then this is one recent discovery, suppressor of gene silencing. That is the host defense mechanism is the gene silencing, and suppressor of gene silencing is something which overcomes the plant defense mechanism. So these are all just the background for you so that we'll be able to follow whatever we have done with plant viruses using bioinformatics tools. Next one please. Okay, before we go into that, briefly, how do we control plant viruses? What I mentioned earlier, natural resistance, by breeding we can incorporate, and engineered resistance. Here is where knowledge of the molecular biology of the virus is important. So we should know which is the gene we are going to engineer and in what way. Next one. And in the engineered plant virus resistance, there are two mechanisms. One is protein-based, where you need to express a functional protein, whether it is a capsid protein or replicase or movement protein, you need to have a protein. Whereas in nucleic acid-based protection, you don't need a functional protein expression. All you need is nucleic acid in the truncated form also, or in the form of capsid RNA. Next one. Now just to give you an example of transgenic protection in tomato, this is cucumber mosaic virus infected, where this is a wild type and this is a transgenic one expressing the code protein genome from cucumber mosaic virus. You can see that the transgenic plants are protected and they give the good yield, whereas the non-transgenic plants are diseased and then they are not able to produce the fruits. loss to the crops. So how do we go about working on these viruses? With the help of bioinformatics. Next one. Okay, this is just again to give you an overall pictorial representation. This is our cardamom mosaic virus. You can see the cardamom leaf with the symptoms. This is the flexuous filamentous particles in EM. This is a soybean infected by the yellow mosaic virus. This is bendy leaf, bendy yellow vein mosaic virus. You can see the geminate particles here. Next one. Okay, now let's start with the cardamom mosaic virus. I don't know how many of you have seen cardamom plants. Have any of you? Okay, good. So you have seen cardamom plants, and this is a mosaic symptom. This is caused by cardamom mosaic virus, which is called as Katte disease in Karnataka. Katte means disorder. So it's the disease is called Katte disease, and it's called cardamom mosaic virus. And when we started this work, there was no information available on this virus molecular biology. Only information was from Cornell University, and they had taken the virus from Guatemala. Guatemala, you know where Guatemala is?
South America.
So Guatemala, not exactly South America, between North and South America, in that region. So that is one of the major cardamom producers of the world. And the Cornell University scientist like Dennis Gonzalez, who was the one actually instrumental in making transgenic papaya against papaya red spot virus. A few years back, he won the World Food Prize because he protected millions of hectares of papaya crops from papaya ring spot virus. So farmers were really happy with that. Okay, so coming to cardamom, he was the one who sent a team of scientists to Guatemala, collected these infected samples. There also the similar virus was existing, maybe it's still existing. So he took the samples, and then they did one electron microscopy study on it, and also serological studies with other panel of various antisera against number of plant viruses. And then they found that it is possibly a 40 virus. 40 virus is 40 viridae, that is a major plant virus family. This is an RNA virus plus stranded RNA virus. Filamentous flexuous particles. So they published a paper, a small paper, long back in plant disease in 1986 or so, saying that this Guatemalan cardamom virus is a possible possible 40 virus with flexuous filamentous particles, and it reacts with some of the 40 viral antisera. And that was the only report. We were wondering why they did not do complete molecular characterization. And then I joined NKU in 1992 and then started working on this cardamom mosaic virus somewhere in the 97 or so, because there was a report in the science section of the Hindu newspaper. Every Thursday or so you will get. There was a report on cardamom virus, not cardamom mosaic virus. There was another virus called Nilgiri necrosis virus. So I contacted the scientist who was working on that, and then characterized the Nilgiri necrosis virus. But then we couldn't get enough sample after that, after the initial characterization for molecular biology work, because that was found only in the Nilgiri region in this one plantation. And since it was a very damaging virus, they destroyed completely those infected plants. So after that, we could not get samples. But then came to know that this cardamom mosaic virus is a major problem in the Western Ghats and all the plantations, and it is not a very serious disease. And because it's not a very serious, I mean, it causes a lot of crop loss, but in terms of fatality, in terms of killing the plant, it was not killing the plant as such, but it was reducing the yield year by year, and it takes some 4 to 5 years for the plant to completely stop producing. But because it was not such a major thing, it was not immediately removed. At the same time, it served as a source of inoculum for spread of the disease. So if you see the, I'll show you later on, if you see the that we want to work on this. Then we got onto collecting this virus sample from the Coorg area and also the Kerala area model. Then we started working on it. Initially we had to do ultrasonography to purify the virus. And then, but we didn't know how to get to the genome. We need to isolate, purify the viral genome in order to clone and sequence it. But we didn't know how to go about it. And that was the time I came across this Gonzalez paper where they had simply mentioned it's a possible member of quadrivalent. There's no other molecular information, only some serological and electron microscopy. And this is where bioinformatics came to our rescue. Because when they had mentioned that it is a possible multivirus, then I went to the database and searched all the multivirus genomes and aligned them. Then found that, next slide please. Yeah, that 40 viruses code for one, the dark line in the center, that is the genome, and surrounding what I have shown is the protein. It codes for one large polyprotein, and then this polyprotein is processed by this protease called NA protease and hexapronate. Okay, that's not relevant now. But what is relevant is that it has a poly A tail. So the moment you see there is a poly A tail, you can make a reverse primer with oligo-DT. Oligo-DT can bind to this, but then we need a forward primer also. So by aligning the sequences of the 40 viral genomes, we found that in the NIB region, NIB stands for nuclear inclusion protein B. There is one A and then there is another B. So nuclear inclusion protein B, which is actually an RNA-dependent RNA polymerase of the virus. So when we align this, we found a stretch of some 20 nucleotides which are completely conserved. Not fully conserved actually, but a few bases are different. So we could make a degenerate primer. Sure, all of you know what a degenerate primer is. That in some positions it can have either A or T or G or C. In some it may be only one or two, A or T or C or something like that. So at that position alone you can indicate the degeneracy. So the oligonucleotide synthesizing machinery is geared to put in all the possible bases in that particular position. So you get a mixture of primers, mixture of oligonucleotides. So we could use that as the forward primer. So the NAB region we could use as a forward primer and oligo-DT because that is complementary to the poly A tail. So using these two primers, next slide please, we were able to amplify a 1.8 kb fragment. Remember, this is an RNA virus, so we cannot simply amplify. We need to use reverse transcriptase, make a cDNA, first strand cDNA, and then a second strand cDNA, and then during this process we could amplify the 1.8 kb fragment. And then, next one please, this is what we cloned and sequenced. So this 1.8 kb fragment contained the partial RNA-dependent RNA polymerase sequence, complete code protein sequence, and a bit of the 3' untranslated region. So when we cloned and sequenced, and then aligned these sequences, then we constructed a phylogenetic tree. We found, to our surprise, that this cardamom mosaic virus belongs to a new genus. New genus in the sense it had only two members, Narcissus latent virus and Maclura mosaic virus. These two were the only members known at that time, maybe somebody during the British rule, somebody brought some, actually Narcissus and Maclura, they are ornamental plants. Maybe somebody brought these ornamental plants and introduced it. From that, this is an aphid transmitted virus in insect. So maybe through aphid, it might have got into the cardamom field, cardamom plantation, and then it has spread all over the Western Ghats. So something like a molecular detective story we could build with this, and then this is what we got. Next one please. And then we also went over all along the cardamom tract, and wherever we went we found this virus. So this is all the way from Sirsi in northern Karnataka to the Idukki district in Kerala. Everywhere there is cardamom mosaic virus. So we collected some 8 different isolates and found, next one please, that even the symptoms are quite variable. You can see some very bright yellow and then this kind of whitish symptoms. Next one. Anyway, so these, here we have some 7 isolates. No, 7 only. So these 7 isolates we cloned and sequenced. In fact, while collecting these samples all the way from Karnataka, my first 2 students, Thomas Jacob and Jabba Singh, they went to collect these samples to Coorg district. And when they were coming, they could not come back actually, because that was the time this Veerappan had kidnapped this Kannada actor Rajkumar. So there was a lot of blockade, and they could not, there were no buses running. So they travelled in a police jeep saying that they have this precious sample for their PhD. So they came back and then did this cloning and sequencing, Jacob and everything. So when we cloned and sequenced, we found that, to our surprise, this northernmost part, Sirsi, the isolate from Sirsi, that was coming somewhere in between the Coorg isolates and then the Kerala isolates. We would have expected this Sirsi isolate to be furthermost, if you just consider geographical ones. But then, again, we don't know how the virus spread, whether it spread from Karnataka to Kerala or from Kerala to Karnataka. But it gives an idea how viruses, the planters, by looking at initial stages, we won't be able to make out whether there is virus or not. Now we have the molecular tools, we have ELISA, we have real-time PCR, all that, and we will immediately detect the viruses. But without these tools, people would have traded infected rhizomes without knowing it's actually infected. So when you plant an infected rhizome, some of the growing plants will show the symptoms, and from there aphids can transmit to healthy plants. That's why all over the cardamom tract you find the viral disease. Next one please. So the next virus I'm going to talk about is a DNA virus. It's called bendy yellow-vein mosaic virus. You can see the plant gets stunted, the fruits are reduced, and these are the yellow vein mosaic symptoms. And this virus again was reported in 1924 from India, but until we started working on it, there was no molecular biology information available. And again, next slide please. Except information that it is a possible member of Gemini viridae. So Gemini viridae, as the name implies, made up of geminate particles, that is 2 icosahedrae are fused together. I showed you earlier that model of this Gemini virus. So based on electron microscopy, people had reported that it could be a member of the Gemini viridae family. So again, we went to the bioinformatics tools. We took all the, not all the Gemini viridae, but in Gemini viridae there are several genera. One of the genus is called bigomovirus. So we took the bigomovirus genus and took the genomes and aligned all these genomes, and we found in the coat protein region there are 2 regions which are highly conserved. Again, not 100% conserved, there are some variations. So we could always make degenerate primers. So we made degenerate primers, and we could pull out 550 base pair sequence, which could be amplified from any bigomovirus species. So base pair sequence. And then from the sequence, again by analyzing the sequence with bioinformatics, we were able to make what are called abetting primers, inverse PCR. That is, two primers just next to each other, but they are going in opposite directions. So when we use this abetting primers, we were able to amplify the complete genome. This complete genome is not very large in the case of Gemini viruses, it's only 2.7 kb. So we were able to amplify the complete 2.7 kb genome of the Bendy 11 mosaic virus. And once we have got it, we could clone and sequence it and the A-type DNA or anything. Don't confuse with ADNA, BDNA or ZDNA. Nothing to do with that. This DNA A stands for one component. You can call it DNA 1 and 2. But then people started calling it as DNA A. DNA A is one of the components of the Ricoma virus genomes. It codes for replicase, code protein and transactivator protein and so on. I will show you that later. And when we align the complete DNA A genome of this Bendy 11 mosaic virus, this was isolated from mother egg. When we do that, we find that the closest relative is from Pakistan. There they call it okra. As you know, Bendy is called as okra in the west, but here we call it Bendy or Venega. And since it was reported first from India, it's called Bendy. We said we will call it only as Bendy 11 mosaic virus. Why should we change it like the Westerns? No, we will call it like what we call it. So it's called as Bendy 11 mosaic virus, and they continue to call it okra 11 mosaic virus. But they are the same, and that is the closest relative. The next closest relative is cotton leaf curl virus. This is also from Pakistan. But both these Pakistani viruses were not characterized in Pakistan. They were sent to UK. Many people still do that. They do not work on it here, they just export it to US or UK, and then they do the characterization, and these people get an authorship. But we have characterized it from Madurai, and where we got it from, we have characterized it there. So this is the next closest relative, and as you know, cotton and Bendy belong to the same family. Can you tell me? Malaysia. Same family. So viruses seem to attack. One type of virus seems to attack members of same family. So these are the closest ones. Okay, next slide please. Okay. Now this is the DNA A component I was telling you, 2741 bases. So what I mentioned earlier was, to begin with, so this green arrow going in this direction, this is the viral cells. That's what it's called AV1. AV1 codes for the code protein of the virus. So a bit of this code protein, so this green arrow is the code protein gene. To begin with, we characterized 550 base pairs of the code protein gene. Then we made abetting primers. That is one primer going in this direction, another going in the opposite direction. That is the inverse PCR. So with the help of inverse PCR, we could amplify the complete circle. So again, if you want to do any such thing, you need to look into the bioinformatics. Only then you will be able to analyze the sequence and to design the primers and make sure the primers are proper. That is, they should not have any hairpins inside the primer. Those two primers should not anneal to one another, otherwise you have only primer dimers. And they should have proper annealing temperature. So all that you need to check beta satellite. And when this report came in the journal PNAS, at that time only we had completed cloning and sequencing of the DNA A component, and we were looking for DNA B component. And whatever we did, we tried various PCR approaches, total DNA extraction, enriching the viral DNA, number of approaches. We could not get any DNA B component. And that was the time the PNAS report came about this AZ11 virus having a satellite DNA. So we thought perhaps our virus also has a satellite DNA. But how do we pull it out? So this is where bioinformatics was very useful. Because soon after publishing that AZ11 virus, they also characterized the beta DNA from cotton leaf curl virus. Remember I showed you the cotton leaf curl also in the phylogenetic tree. So we were able to align the cotton leaf curl virus and then the AZ11 virus, and then we found to our surprise around 100 to 120 base pairs were completely conserved, nearly 98% conserved between these two viruses. So we took this region, and then knowing that it's a circular single-stranded DNA genome, again we made abetting primers, inverse PCR we did, and then we were able to get a beta DNA. So, next slide please. Okay. Once you have these two components, the DNA A and the DNA beta component, what we did was make partial tandem repeat clones of both DNA A and DNA beta, and we did what is called agro-infection of pediclams. You notice here DNA A alone produces only mild leaf curl. Beta alone does not produce the symptoms, but if you put A and beta together, you get the typical yellow vein mosaic symptoms, which could be transmitted by the insect vector whitefly to susceptible plants to produce the typical symptoms. This is mock-inoculated vector. This is the naturally infected pediclam. So now we know that the DNA A and DNA beta satellites, they both are necessary for typical yellow vein mosaic symptoms and bending. Next slide please. So this work was done by my second student, Joyce. So we reported this work in the journal Virology. Bendy yellow vein mosaic disease in India is caused by association of the DNA beta satellite with the bigommovirus. So this couldn't have been possible at all without the help of bioinformatics. So I cannot thank bioinformatics enough for helping all our activities. So next slide please. To analyze the satellite DNA, so this is the region which is called the satellite conserved region. It has a stem loop structure, and this has a conserved mononucleotide. That is 9 nucleotides are completely conserved. And then this has an A-rich region, and this is a single ORF in the complementary sense. It's called beta C1 ORF. Next one please. This is another further analysis of this. This is the conserved region, which is GC-rich. Next one please. Okay. Now, what is it that is common between the DNA A component and the beta component? The stem loop structure and the TAATAC, that mononucleotide sequence. So the replicase protein which is coded by the DNA A genome is making a nick in the loop region and helping in the replication of the beta DNA. So beta DNA alone does not have any existence, but it needs the DNA A component and the replicase for its replication. Then what is the role of the DNA beta? What is the role of the satellite DNA? So next one please. Okay, we can skip all these alignments. Next one. Okay, maybe you can skip just to show what is the. So ours was the third report in the world about the satellite DNA, first in India, and very soon, we reported in 2003. Now in the past 8 years, some hundreds of beta DNA have been reported all over the world, especially in China. Next one please. You can see all this CN stands for tomato link, China virus, various isolates, everywhere they find the DNA beta. Just to show you what is the influence of bioinformatics, you can see this is the ORF and our virus, interestingly, when the 11 virus, it has an extension to the endovirus in the beta cell. Again, this is the collagen tree based on this beta-11 virus. Next one. And we found that DNA A and the DNA beta, they seem to have co-evolved. Next one. Now we know that there is a DNA A, there is a DNA beta, which is half the size of the DNA A. But what is the functional relationship between these two components? Next one. Okay. So for this, we launched into another project, and then again we were able to take only the beta C1 ORF and we made transgenic plants, Nicotiana, Benthamina, in the sense orientation and in the antisense. And you notice here, the sense transgenic plants are completely deformed, whereas antisense transgenic plants are not affected at all. That means the protein which is encoded by the beta C1 ORF, that is essential for the symptom development. We have also shown in this paper, in the write-up I have given all the references, so if you are interested you can read it. That is, the beta C1 is involved in suppression of post-transcriptional gene silence. Next one. Okay, we have also tried using molecular modeling to find out what are the surface exposed regions of this spot protein. So it's a monomer and this is a pentamer. This is an icosahedron, so it can assemble as pentamers. Next one, please. Okay. We know that it is a bigoma virus, Bioinformatics virus and so on, but what is the origin of the beta DNA? How did the beta DNA evolve and how is it able to contribute to the symptom development and thereby the pathogenicity? For this, we went around all the bendy fields and found that these are all the weeds which are present near the bendy cropping fields like cedar, croton, malvastrum, vernonia and HLS. So we went around amplifying these genomes, DNA A component, and some of them had the DNA beta component also. So we cloned, sequenced and then compared it to the beta DNA of the bendy 11 sequence. To our surprise, none of this beta DNA is homologous to the bendy 11 sequence beta DNA. So still the search is going on. Where did it originate from? But in next one, in this process, we hit upon this Vernonia elevine virus, which turned out to be a distinct bigoma virus species. One day I got an email from the International Committee on Taxonomy of Viruses saying that you have discovered a new species, nothing but Vernonia elevine virus from the common weed Vernonia cinerea. You can see this symptom looks very much like the bendy virus, but again it's not homologous. In fact, it's not homologous to any of the existing bigoma virus. It's a new species. So my student Bhagyalakshmi has done this. Next one, please. Okay, now coming back to the earliest record of plant viruses, I told you, this is the poem. Can anybody read Japanese? Here?
I can speak little Korean, not Japanese.
So, this poem is what I showed you earlier, no? How is this plant showing yellowing in summer? It's supposed to show yellowing only in the autumn season. But this is the poem, Empress Koken. I showed you earlier that this earliest 750 AD was the earliest record of a plant virus disease, without knowing it was a plant virus disease. This plant is an ornament of plant called Eupatorium. The disease is Eupatorium elevine disease and virus is Eupatorium elevine virus. Now why I am showing this again is, this virus also has a DNA beta satellite. And again this virus was taken from Japan and it was characterized by Saunders and John Stanley in UK. Again in John Innes Institute, and they published it in Nature, saying that characterize the earliest plant virus. But what is interesting is this virus and our bendy virus, they both have the beta DNA and they belong to the same group of viruses, that is monocotic viruses requiring a DNA beta satellite for disease symptom pathogens. So that way we are quite close to the earliest plant virus record. Next one. In fact, this bendy virus has put us on the international map. Like you can see, two of the viruses which are recorded for India, that is the bendy 11 mosaic virus, and we also characterize the Asiatum 11 virus from India. They both require, but this is quite old, 2003. Recently in 2010 only we published that Vernonia 11 virus, so that also should come here. The third one here is cotton leaf quill virus, which is present in Haryana. Cotton leaf quill is a major epidemic. It wiped out a lot of cotton fields in Pakistan, and it has spread to areas like Haryana, Punjab and all. So people in IIS Bangalore have characterized the cotton leaf quill virus. So that is the third circle here. So this came as a review in Trends in Plant Science. Next one please. Okay, now coming to the third virus, third crop we are working on is soybean. As you know, soybean is a very rich protein content, rich in protein. That's very good for vegetarians, of course, for everybody. And India is the fifth largest producer of soybean in the world. Unfortunately, this soybean is highly susceptible to the yellow mosaic virus. You can see that this whole field is infected with yellow mosaic disease. This is a field in Madhya Pradesh, in Jabalpur. So this is where we got the virus sample from. Next one. This is a typical Bicoma virus. Does not have a satellite DNA, it has DNA A and DNA B, transmitted by white fly. This is a germinate particles. Next one. We characterize this virus both from Madhya Pradesh and from Madurai. And you can see that when you use, this is a naturally infected, when you use DNA A alone or the B alone by partial tandem repeat construction and do an agroinfection, you do not get any symptoms. But if you put both A and B together, you get the typical yellow vein symptoms. So we showed that this is indeed the virus that is causing the yellow mosaic disease in soybean. Next one. Okay, this is the genome components, and this is the paper. Student Girish did this work in published in Virus Research. Again, analyze all the genomic components, getting the ORFs, and we have also characterized the nature of this protein, which is given here as PC1, which is coding for cell-to-cell movement of the virus. So we have characterized that virus protein. We have expressed the protein. Again, in order to do all that, we need to analyze the genomes, and again to analyze the genomes we need bioinformatics. So analyze the genome, we have cloned this region which is shown in blue here, and then shown what is the mechanism involved in the cell-to-cell movement. I am not going into those details. But again, I am emphasizing it is important to have a knowledge about the genomes and then go into the proteomes. And also we have characterized this region, what is shown here in blue, it is AC1, that is the replication associated protein. We have taken this and then expressed it in E. coli, and this protein we have purified and then shown which is a specific nucleotide it is binding, because this rep is the one which makes a nick in the rolling circle replication mechanism of this. This is single-stranded DNA. So just like the bacteriophage and 13 and some of the 5x174, all those it's involved in single-stranded DNA replication. So that we have shown using the bioinformatics tools. Next one. Here is what I want to point out something. So here you notice this here. Let's concentrate on this green arrow here. Let's concentrate on this green arrow here. Written as MYMIV-SBMT. That is mung-bean yellow mosaic India virus from Madhya Pradesh. The soybean, there is so much sequence divergence. They belong to two different species. One is MYM-IB and another is MYM-MD. So we have characterized both. This MYM-IB SB-MD is from Madhya Pradesh, Jabalpur, and what they call as MAD. Maybe looking at this, but actually it stands for Madurai. MYMB, SB, MAD. So these two are the two species we have characterized. And then I'll come to this later, that is MYMB HGTN. This is the original name, that is Mongolian yellow mosaic virus Hosgram. Next one please. Actually, this also turned out to be a new species of bigama viruses. How do we say it's a new species? In Gemini viruses, there is a rule that if the DNA A component has less than 89% identity with the existing virus, then you can call it as a new species. That is how we got the Ergonia 11 virus. And to our surprise and delight, we got one more new species, that is Hosgram yellow mosaic virus. And I'm sure people here will be happy to note COI is added. COI stands for Coimbatore. Because we got this Coimbatore isolate from TNAU. So I must thank Pulses department, Dr. Kumar Varivel and others who have given us this sample. This is the Coimbatore isolate. India, Coimbatore. So this is the Hosgram yellow mosaic virus. Again, this is a new virus causing Hosgram yellow mosaic disease in India. This was characterized by Dr. Barnabas. He is associate professor in the American College in Madurai. Again, he has shown that the DNA A component and B component alone do not cause the disease, but when you put them together, you get the typical yellow mosaic symptoms. So, but then the legume viruses themselves form a separate phylogenetic tree. And in the phylogenetic tree, we find that MYMB and MYM-IB are forming two separate clusters. And the Hosgram yellow mosaic virus stands out. So because it's a new species. So all this is possible with bioinformatics tools. Next one. Now what is the application of plant virology and functional genomics? Next one. So with the large number of sequence information available in the databases on plant genomes, we have to find out what are the functions of this. So for that we need some tools, and we have developed some tools using both the soybean virus and the Bendy virus by replacing parts of these viruses with genes from the plant, the host genes. Next one. That is called virus-induced gene silencing vectors. So, next one, this I have shown already. Next one, please. Previous one. So the idea is, you replace part of the viral genome with genes from the plant. Not even the complete gene, some part of it, because that part of the sequence is enough for triggering the gene silencing process. But it is induced by viruses. So it's called virus-induced gene silencing vectors. So when we have demonstrated this with this phytoin desaturase, it's involved in carotenoid biosynthesis. So if you silence this gene, you will have a bleached phenotype, because the plant is not protected against the UV light. So when we put this beta C1 ORM, replace it with gene from the plant, we see that this bleaching is reduced. So we can use this vector for finding the functions of the plant genes. We can take the cDNA library from the plant genomes, insert those genes into our mixed vectors and infect plants, and based on the changes it produces in the plant, we can find out the functions. Next one. This is again to show when you clone GFP into this mixed vector, how it is functioning. So if you take a GFP expressing plant here, this one, and then if you infiltrate with our mixed vector carrying GFP, then GFP is silenced. That is, the idea is to silence an endogenous gene. So in this case, already there is a transgenic plant expressing GFP, But if you introduce that using your virus-induced gene silencing vector, then it will be silenced. Next one. Okay, the last part of my talk will focus on applications of plant viruses and even developing vaccines. So now you can see this is a chimera. You have a rabbit body and a lion's head. That is hybrid. So the chimera actually stands for, it's a Greek word where lion's head, goat's body and a snake's tail all put together is called a chimera. It's an imaginary animal, something like what we have in our own legends. No? For our Ganesha, no? We have an elephant head and a human body, or Kamadhenu with a woman's head and a cow's body, say things like that, or mermaid. So something like that. What we are trying to do is, using the plant virus body, we are engineering epitopes from animal and human viruses. So next slide. Okay, so this can be used for molecular forming using plants as the factories. Next one. Okay, now we are coming back to our cardamom mosaic virus. Earlier I showed you how we characterize the cardamom mosaic virus. Next one. We have also expressed the core protein gene of the cardamom mosaic virus in E. coli, and we have shown that it assembles like filamentous particles. Next one. So using this, we are expressing epitopes from HIV. So the GP41, there is a conserved region which gets exposed during infection. Next one. This is just to show you these regions, that is what we denote as E1 and E2. These are the regions which form major epitopes in the GP41 protein of HIV. So the idea is to express these epitopes on the surface of the plant virus core protein. Next one. Okay. To begin with, how do we go about it? Again, we rely on bioinformatics. We go to the database and find out what are all the HIV isolates which are reported from India. Here again, we found wherever I have simply written India, these are the ones which have been sent abroad and they have sequenced it. But some of the viruses we do have information from India, done in India itself, that is the National AIDS Research Institute in Pune, and then the Jawaharlal Nehru Center for Research in Bangalore. There they have done the complete sequencing of the HIV genome. So we aligned it and then found a consensus sequence. Next one. This is the other region, what I call E2. Here is where we found the Bangalore isolate. So this contains these two epitopes which are highly conserved. So we have a DBT project on nanoscience and nanotechnology where we have expressed these epitopes on the surface of the cardamom mosaic virus protein. So this virus, when it is expressed in E. coli, it assembles like a large filament. So we have multiple copies of this epitope on its surface. So up to 2000 copies it will form on the surface of the virus-like particle. So this can be used as an effective candidate for vaccine production. We have a collaboration with the Tuberculosis Research Center in Chennai, where the scientist Professor Ramanathan, who is involved in the International AIDS Vaccine Initiative. So he is collaborating with us. So we are able to get some sera from HIV patients. So some of these sera, we have tested so far some 90, out of which some nearly 10 of them are interacting with our protein, this plant viral protein expressing the epitopes from HIV. Again, we couldn't have done it without the help of bioinformatics. First of all, we should know which region to engineer and then which isolate to take. We have to take all the Indian isolates, align them and then see whether it is conserved or not. And then also on our plant viral protein, we should find out which is the region where we can engineer. Because the assembly of the viral particles should not be hindered by expression of foreign epitopes. By steric hindrance, it should not be able to form the particle. So we need to analyze all that these are the regions which have a lot of random points, do not have any stable secondary structure. So it gives us a clue that these regions are highly surface exposed. So these regions we chose for expression of foreign epitopes. So we have some positive evidence. So we hope this will come out to be at least a diagnostic kit for detecting AIDS. Next one. Okay, so we are working on plant viruses with the help of bioinformatics. We can go from plant virus molecular biology to structure to vaccines. Now I would like to acknowledge all the students and postdocs and MSc students who have worked on these viruses. Like Thomas Jacob was the first student, then came Archana Jagga Singh, and then a number of postdocs and project fellows who help. And then currently Shubha and Karan Shelby are working on the HIV epitope presentation on cardiac mosaic virus. Then for the Bendy virus, Joyce was the first one to work on the molecular characterization. Then later on Praveen, Gopal and they worked on the characterization of what are the functions of the various viral proteins. Now currently Jaya Bharati and Sridharan, they are working on the virus-induced gene silencing vectors. Then soybean virus, Girish, and then Oscar-Mello mosaic virus, Barnabas, and then the Vernonia virus like Bhagilakshmi and the Vernonia virus, and a number of other viruses. We have a project also to make transgenic soybean resistant to a number of isolates. So again, the movement proteins are highly conserved, so we are making transgenic soybean with movement protein genes. So, next one please. Finally, thank you. These are all some of the birds from our campus. And I would like to thank the Bioinformatics Center of NKU, where we could do all the sequence structure analysis. And then funding. Without funding, we can't do anything. So, CSAR was the first project, then DPT, DST, and then UGC. I'd like to acknowledge the funding from all these agencies. Finally, thank you once again for